Okay, so here's the assumptions part. So you're going to start back with, okay, so we've got the Hardy-Weinberg equation, p squared plus 2pq plus q squared. And using that equation, we can predict what the next generation is going to be. If we have a bucket of interbreeding little organisms, their gene pool, we can predict the next generation over and over and over again. So the question becomes, then why do populations actually change? Because remember, we initially defined evolution as the change in these allele frequencies, the change in the number of yellows and whites over time. So do populations ever actually change their allele frequencies over time? And obviously the answer is yes. We've seen that evolution does occur. So the Hardy-Weinberg equation, how useful is it? Is it actually predicting anything? So that's what we're going to look at now, is what happens to actually change allele frequencies. Because we've seen that over time, just by mating over and over again, that does not change the allele frequencies. Just being dominant does not change the allele frequencies. So why do allele frequencies change over time? Why does evolution occur? So we're going to use this population of snails. So we can read this little beginning part. We've got a species of Littorina saxatilis. It's a little tiny snail that lives on the rocky outcroppings that go down into the ocean. And they can live at 3 different levels. They can live at high tide, where they almost never get wet. They can live at mid tide, where they get wet as the tide goes up and down. Or they can be at low tide, where they're almost always submerged underwater. And these snails live at these 3 different populations. Well, scientists went in there and they looked at a particular allele, and it's for a metabolic enzyme. It doesn't matter that they know this, but it's a particular metabolic enzyme that they're looking at, and they looked at, it has 2 different alleles. Okay, and we're going to label those alleles as the yellow allele or the white allele. All right, so scientists went in and they looked at these snails. This is based on a real story. And they looked at these snails, and this is what they found, that at high tide, 90% of the alleles were this yellow one. At mid-tide, 85% of them were yellow, and at low tide, only 10% of them were yellow. So that's kind of weird. So say, go ahead and do number 1. Right? Number 1 asks, with your group, come up with hypotheses to explain why the groups might have different allele frequencies. Oh, and by the way, the gene is called the AAT gene. So that's in that number 1. It says the AAT gene. So we're looking at the AAT gene. Just the name of this particular enzyme. Okay? Some kind of amino acyl transferase, I believe. All right, so anyway, so they're going to sit and think about hypotheses, okay? And it can be anything. There's no right or wrong answer for this, but go around and talk to the groups and ask them what they're thinking. Maybe there's a predator that lives in the water that finds the yellow allele or the white allele really tasty. No, the yellow allele really tasty. And so they're more likely to have the white allele down here. Or maybe the yellow allele is better for hot, dry temperatures, and so if you're above the water and not getting wet all the time, the yellow allele is better. Maybe there's different food types that they feed on at high, medium, and low. And so one allele is better at digesting one, and one allele is better at digesting the other, and so they have to be there. Or maybe the coloration of the snail matters. So if they're underwater, they have to blend into the seaweed and the moss and stuff, and so they're a different color. And maybe this AAT allele or gene controls color, whereas when you're above it, you need to be more of a gray color so that you blend into the rock. So there's a million different reasons that they can think of. Just have the groups share those reasons. There's no right or wrong answer yet. All right, so they're just going to share out reasons that it might be. But then emphasize that, do you think that these guys are all one breeding population? Are they one breeding population? And we say, are they one population in Hardy-Weinberg equilibrium? Meaning that you could use the Hardy-Weinberg equation to predict the next population every time. Excuse me, and it should be very obvious to you that no, they're not. These ones maybe a little bit, but this one's definitely not the same population. It's like having this, right? Okay, so they're not in the same population, or they're not in the same frequencies. And so one of the things you might ask is, well, why don't they just, if they're always mixing, why don't they kind of even out their proportions? Because if I dumped these 2 buckets together, we would certainly come up with an even population. So students should be able to say that, well, they're probably not mating with each other, right? They're not interbreeding with each other. And so that's one of the things you can say, oh, so something that doesn't allow them all to be in Hardy-Weinberg equilibrium together is if they don't mate randomly. Okay, so in order for them, let's see, this is so we're going to do Hardy-Weinberg assumptions. So in order to stay in equilibrium, so that's what you would say, to stay in equilibrium, i.e. to not evolve. Number one, you have to have random mating. They have to all be mating with each other and not doing this little niche non-random mating amongst their own little populations. They have to be randomly mating. So that's one of the assumptions we make for Hardy-Weinberg is that they're randomly mating. There's no non-random mating. They're all mating randomly. Okay, so another way to state this, well, we'll do that in a minute. Okay, so no non-random mating. All right, so then in number 2, let's see. Number 2 asks them, why might these populations remain as 3 distinct populations? Oh, and that's where you're going to bring up the non-random mating and the fact that the environments must be different, okay? So we can't have different environments. If we have different environments, we're going to see evolution. So we have to have no environmental change. Oh my gosh, I can't spell. Environmental change. Okay, so no non-random mating and no change in the environment. Otherwise, you're going to have differences in populations. All right, so then go ahead and have them answer the next part. So it says, in 1988, a toxic algae kills off most of these guys, but these guys remain intact, okay? Oh, and by the way, sorry, so you're going to assign, these 2 tables as low snails, these 2 tables as mid snails, and those 2 tables as high snails, and whatever you want for the last one. They're going to be using these bags, right, that represent the high tides that are about 90% yellow, the mid tides that are about 85% yellow, and the low tides that are about white percent yellow. White percent, 10% yellow. Okay. Now these are going to get sort of mixed up and screwed up during your lab, so I'm sure that the TA of the next lab would really appreciate it if you would try to sort them back out into these approximate percentages at the end of your lab, or have students help you do that. All right. So they're going to each get a little cup, about 2/3 full, of one or whichever population they are.